So, Joanna, when you...
Okay. Good afternoon, everyone. So it is my great pleasure to introduce you our speaker of today, Dr. Águeda González Rodríguez. She's a senior scientist at the Sols Morreale, the Biomedical Research Institute in Madrid, Spain, and she will deliver a seminar today entitled The Inhibition of ALK3-mediated signaling pathway protects against acetaminophen-induced liver injury. Dr. Águeda, please go ahead.
Thanks for your kind presentation. Good afternoon, everybody. And thanks the organization for giving us the opportunity to explain our last paper published regarding acute liver failure and BMP proteins. Bone morphogenetic proteins are growth factors belonging to the transforming growth factor beta family. They were firstly described as inducers of osteogenesis and regulators of bone structure formation during embryogenesis and as well as fracture repair in adults. Despite receiving their name from their capacity to induce bone formation and remodeling, their involvement in the development and function of different organs and systems is currently known. Indeed, they are also involved in processes of organogenesis and embryogenesis, apoptosis, chemotaxis, and repair of a wide variety of tissues, as well as in glucose and iron homeostasis. At present, more than 20 BMP proteins have been described. BMP proteins show homology to each other and to other TG-beta family members in the amino acid sequence. Regarding their signaling pathways, there are two types of serotonin receptors involved in the signal transduction induced by BMPs. Type 2 receptors are constitutively active and can recruit and phosphorylate type 1 receptors, including ALK2 and ALK3, which are the subject of this study. Regarding the liver, previous research have demonstrated that while both receptors are expressed in human liver and all hepatocyte-derived cell lines, other type 1 receptors, such as ALK6, was not detected. Although BMP ligands can bind to either type 1 or type 2 receptors, for signal transduction, ligands have to form a complex with two subunits of type 1 receptor and two subunits of type 2. Once the complex ligand receptor is formed, the receptor subunit transphosphorylates and become active. The activated complex phosphorylate SMAD158, which binds SMAD4 and translocates to the nucleus, inducing the transcription of target genes. Among this canonical pathway, there is a non-canonical signaling, which is independent of SMAD proteins mediated by other kinases, such as PI3 kinase and MAP kinase, can also transduce BMP signaling. Given the larger number of existing BMP ligands versus the low variability of receptors, different degrees of affinity of each ligand for different receptor types have been observed. Generally, this affinity is shared among members of the same BMP protein subfamily, as it depends on structural elements and exposed residues in the binding sites of both receptor and ligand. As mentioned above, ligands can bind either to the already formed receptor complex or to type 1 or type 2 receptors separately. Thus, BMP2 and BMP4 preferentially bind to type 1 receptors, having higher affinity for ALK3 and ALK6, and subsequently requirement of the type 2 receptor. Of course, in contrast, members from OP1 family, such as BMP5, 6, and 7, bind first to the type 2 receptor, followed by binding the type 1, with their higher affinity for the ALK2 receptor. As previously mentioned, the osteogenic capacity of BMP proteins has always been their main characteristic. However, it is now widely known that these growth factors exert pleiotropic effects in different tissues through different physiological processes. So a large of pathologies have been associated with defects in the BMP signaling pathway. In addition to bone disorders such as fibrodysplasia or arthritis, BMP have been also associated with diseases unrelated to the skeletal muscle system, such as multiple sclerosis, as well as respiratory and vascular diseases. Similarly, some components of the BMP family have linked to certain types of cancer. Moreover, the role of BMP proteins in the progression of renal fibrosis have been largely studied, and recently some BMPs have been described to be involved in the function of adipose tissue by regulating adipogenesis and thermogenesis. Regarding the impact of these proteins on liver physiology, it is known that they play a crucial role in different stages of embryonic development of this organ, and that they are also involved in the homeostasis of the liver in the adult stage. Acute liver failure is defined as the development of hepatocellular dysfunction in patients without resistant liver disease with 7 to 26 weeks. The key event in the development of acute liver failure is a massive hepatocyte death that overwhelms the capacity of liver regeneration. It can manifest as coagulopathy and encephalopathy, in addition to jaundice and reduced metabolic capacity. The causes of acute liver failure widely vary depending on socioeconomic factors, exposure to risk factors, and other genetic factors. The most common cause of acute liver failure, especially in Western countries, is drug-induced liver injury, including paracetamol overdose, following by viral hepatitis and vascular and metabolic disorders such as Wilson's disease. As just mentioned, the main cause of drug-induced liver injury is paracetamol overdose. This drug, also called acetaminophen in North America, referred as a PAP in this presentation, is an analgesic and antipyretic drug widely used worldwide. Even though it is a highly safe drug at a therapeutic dose, intentionally or accidental overdose can lead to acute liver failure. Indeed, a single dose between 10 and 15 grams of a PAP can exert fatal effects on the liver, triggering massive hepatocyte necrosis. Especially hepatotoxicity occurs due to an imbalance in hepatic APAP metabolism. A glutathione depletion caused by the APAP overdose or low reserves of this peptide due to different systemic conditions, including fasting or alcohol consumption, results in accumulation of the highly reactive species NAPK derived from APAP metabolism through the enzyme CYP2E1. Producing NAPK forms covalent bonds with mitochondrial proteins in hepatocytes, interfering with the electron transport chains and producing reactive oxygen species, ROS. Eventually, DNA fragmentation occurs, and consequently a massive death of the hepatocytes is produced. The rapid progression of acute liver failure from acute liver dysfunction to multi-organ failure makes early diagnosis and treatment necessary when making decisions, such as the susceptibility of each patient to undergo hospitalization or liver transplantation. Nowadays, diagnosis for APAP-induced acute liver failure is based on the symptoms of the patient, classifying between liver failure caused by APAP intoxication on all other causes. In recent years, interest in the search for specific or non-invasive biomarkers for diagnosis and prognosis has grown notably. Currently, the only biomarker that has been proposed for diagnosis of APAP-induced liver failure is the APAP protein-derived cysteine adduct. Regarding APAP overdose, N-acetylcysteine NAC is the standard treatment and it exhibits high safety and is highly effective. However, it must be administered within the first 8 hours post-ingestion of a high dose of APAP. So it presents a very narrow therapeutic window. This drug is a precursor of glutathione, so it restores its reserves in the liver. The increase in glutathione stores favors the metabolization of the reactive NAPK and counteracts the ROS production in the mitochondria. Also, gastric lavage, which activates charcoal, is also recommended in patients whom intoxication is detected within the first 4 hours, due to it reduces APAP absorption by 50 to 90%. Sometimes liver impairment is so severe that recovery is not possible. In this case, the definitive treatment is liver transplantation. There are also artificial support systems, liver support systems that are useful to optimize hemodynamic, neurological, and biochemical parameters, either prior to liver transplantation or to facilitate the recovery of the patient. So far, there are not many investigations focused in the involvement of BMP and their signaling in the progression of acute liver failure. However, the role of BMPs in hepatic iron homeostasis have been extensively studied. Synthesis of hepcidin, a key molecule in the regulation of iron metabolism, is induced by BMP2 and BMP6 that signal through the type 1 receptors ALK3 and ALK2 respectively. Some studies have shown the association between these BMP proteins with diseases that involve iron overload, which may be the trigger for acute liver failure. In fact, in a study focused on BMP proteins in drug-induced liver injury, it was observed that mice subjected to an APAP overdose presented an increase in the concentration of hepatic iron. This study suggests that the oxidative stress produced by excess APAP results in the inhibition of hepcidin synthesis by a blockage of BMP signaling pathway. On the contrary, in a recent study, an induction of the BMP signaling pathway was observed in mice with an APAP overdose model. Therefore, there are contradictory results regarding the activation or inhibition of BMP signaling pathways in this context. Taking all this background into account, the aim of this study was to determine the role of BMP protein signaling, particularly the BMP type 1 receptors ALK2 and ALK3, in APAP-induced liver injury, and to assess whether pharmacological targeting on these receptors might have a beneficial effect in the treatment of acute liver failure caused by APAP. In order to explore the role of BMP signaling in APAP-induced acute liver failure, we use several pharmacological inhibitors which present different selectivity for each receptor in a human hepatoma cell line A2A7. For example, we use DMH2 to selectively inhibit signaling through ALK3, ML347 for ALK2 specific inhibition, and LDN 193189 to achieve the inhibition of both receptors. After exposure to these inhibitors, lower phosphorylation of SMAD158 was observed for all of the inhibitors, evidencing an efficient blockade of the signaling pathway. Curiously, we observed a modulation in ALK receptors expression in cells treated with DMH2, a decrease in ALK3 and upregulation of ALK2 expression. In addition, we quantified the expression of well-known BMP signaling targets, ID1, which codifies for DNA binding protein inhibitor ID1, and HAM, which codifies for epsilon, finding that the expression of the latter was reduced in those cells where ALK2 was inhibited, while ID1 expression was more significantly decreased in cells treated with those compounds inhibiting ALK3. Given that epsilon gene expression seems to be more influenced by signaling through ALK2, while ID1 expression by signaling through ALK3, we used these two target genes to further validate the activation of this receptor in our cellular system. Next, HU7 cells were treated with the inhibitors prior stimulation prior to APAP stimulation, finding that in the inhibition of ALK3 achieved by DMH2 ranked higher cell viability after APAP exposure, and this protection was not achieved by using any of the other test inhibitors. For evaluate if these results were recapitulated in primary mouse hepatocytes, cells were treated with different doses of DMH2 in order to inhibit ALK3 signaling prior to APAP exposure. The results show the higher viability after DMH2 treatment compared to non-pretreated hepatocytes. Since similar effect on DMH2 in primary hepatocytes and in HU7 cells was found, we used these cells, the cell line, for further experiments. Well, as higher cell survival was observed in those cells pretreated with DMH2 compared to the corresponding controls, we performed assays of cytotoxicity and apoptosis to deepen into this finding. We observed a reduced APAP-induced toxicity reflected in less release of LDH, a well marker of necrosis, as well as in decreased processing of caspase-3, a marker of apoptosis. In addition, it is well known that an antioxidant response is triggered in order to avoid APAP-induced oxidative stress. Thus, RNA levels of antioxidant genes were measured as indicators of the activation of this antioxidant response. As expected, an induction of these genes was observed after APAP treatment. However, no increase of these antioxidant genes was detected in DMH2 pretreated cells. Also, it is well known that cross-production triggers stress kinase signaling. So, according to the observed results, APAP-stimulated cells pretreated with this inhibitor saw lower phosphorylation of the stress kinases JNK and p38 after APAP treatment. To further corroborate the implication of DMP type 1 receptors in APAP-induced acute fever damage, we performed a short high-pain knockdown of ALK2 and ALK3 in HUH7 cell line, achieving a significant reduction of both receptor expression. We also observed a lower phosphorylation of SMAD158 in silenced cells. Curiously, we found that ALK3 expression increased due to ALK2 signaling, and that ALK2 expression also was elevated in ALK3 silenced cells, similar to the modulation observed after DMH2 treatment. We also observed a modulated expression of the target genes. As observed in the previous cellular model, hepcidin gene expression decreased in ALK2 silenced cells, while it increased in cells with a mild overexpression of this receptor. On the contrary, ID1 expression was reduced after silencing of ALK3, and it was elevated in cells with a mild overexpression of this receptor. So again, hepcidin gene expression seems to have been more influenced by signaling through ALK2, and ID1 more influenced by signaling through ALK3. Once established the silent cell lines, they were treated with APAP, and cellular viability was assessed, showing an increase in cell survival in ALK3 silent cells after APAP compared to the controls. This effect was not observed in the cell lines with ALK2 signaling. Moreover, we detected a lower release of LDH, as well as a lesser amount of the cleavage fragment of stage 3 in ALK3 silenced cells treated with APAP compared to the control ones. Furthermore, we use DHE proof to measure ROS production in these cells during the 2 hours after APAP stimulation, and we found that APAP-induced ROS was almost not observed in cells silenced ALK3. Interestingly, a higher expression of the antioxidant genes at baseline and lower expression and lower increase in the expression of these genes after APAP treatment was observed in ALK3 silent cells. Finally, lower levels of phospho-JNK and phospho-p38 were found in ALK3 silent cells compared to control cells. As stated above, ALK2 expression was significantly increased in both ALK3 silenced cells and DMH2 pretreated cells, possibly by a compensatory mechanism against the absence of ALK3 activation. In order to explore the implication of increased ALK2 expression, we infected 8 UH7 cells with lentiviral particles to generate a stable cell line expressing ALK2. The overexpression of ALK2 was evaluated by measuring RNA levels of this receptor, which show a 3-fold increase expression. Also, the expression of its target gene, hepcidin, was significantly increased, while ALK3, and ID1 expressed were not modulated. After stimulation with APAP, cells overexpressing A2 did not show higher cell viability. However, a strong inhibition of the cleavage of KSP3 was observed, indicating that ALK2 overexpression displays an anti-apoptotic effect. On the contrary, no difference in ROS-induced antioxidant genes and in the phosphorylation of stress kinases was observed. Well, in an attempt to explore whether the effects observed after ALK3 inhibition in a PAP-induced toxicity in the cellular system could be reproduced in vivo, we performed an experimental model of a PAP-induced acute liver failure in mice. Since we wanted to evaluate the potential effects of the administration of DMH2 on liver damage after a PAP overdose, we intraperitoneally injected mice with a vehicle or a PAP, and 1 hour later with DMH2 3 milligram kilo, or the corresponding amount of each vehicle was administered. In order to assess the effect of this inhibitor on a PAP-induced liver damage, we quantified the percentage of injured areas in the liver tissue of these animals. Notably, a significant reduction of a PAP-induced hepatocyte damage was observed only in those mice treated with DMH2 after a PAP challenge. Likewise, serum ALT serum transaminases levels after a PAP-induced liver injury were significantly lower in mice treated with DMH2 than in those receiving with the vehicle. In addition, tunnel staining revealed less presence of pyknotic nuclei in the livers from animals treated with DMH2. Indicating that this inhibitor protects against a PAP-induced apoptosis in mouse livers. As previously mentioned, one of the main features of a PAP-induced liver injury is the production of ROS and the triggering of the antioxidant mechanisms in response to oxidative stress. In order to assess the effects of DMH2 treatment on a PAP-mediated oxidative stress, we measure the hepatic expression of several antioxidant genes. As expected, a marked increase of antioxidant gene expression was observed in liver samples from mice exposed to a PAP overdose, but this decrease was milder or was not occurs in the presence of DMH2. According to this data, treatment with the inhibitor also significantly reduced a PAP-induced glutathione depletion. Okay, to further validate this finding, we quantified the hepatic expression of 4-HNA, a secondary product of lipid peroxidation, finding that it was significantly lower in livers treated with DMH2 than in those treated with vehicle. Finally, and as expected, lower levels of phospho-junk were found in DMH2 treated mice. For the evaluation of later time points, we have analyzed livers from mice that have been sacrificed 6 hours after DMH2 post a PAP treatment. Although we found a protection from a PAP hepatotoxicity, it means DMH2 reduces necrosis and apoptosis and lipid peroxidation. induced by a PAP in this model, this effect was not as significant as at 2 hours after a PAP treatment. So indicating that an attenuation of the effect of the inhibitor at longer time periods. Okay, taking into account this study demonstrated that the inhibition of ALK3 protects against a PAP-induced hepatotoxicity, providing new mechanistic evidence on the role of BMP signaling in the pathogenesis of liver injury mediated by a PAP. These experimental findings open up a novel therapeutic approach for patients with acute liver failure due to a PAP overdose, and therefore clinical studies to assess safety and efficacy of targeting BMP signaling in patients with this life-threatening disorder are warranted. And finally, I want to thank people from the lab. This work was performed in the Laboratory of Metabolic Syndrome and Vascular Risk at the research unit in the Hospital Universitario La Princesa Santa Cristina, where I was working until I moved to the IBE Sols Monreale. And I want to thank all the collaborators. And now I'm ready for your questions.
Thank you so much, Águeda, for your very interesting talk. And I give the opportunity for the people that is attending the seminar to make some questions. Bernard, I see that you already...
Thank you very much, Joanna. Yes, thank you, Águeda, for your very nice investigation in vitro and in vivo. It's a very new results and new idea for those who are working on this drug, very, very important drug in pharmacology. My first question is whether you had the opportunity to evaluate the expression of CYP2E1 or the activity of CYP2E1 in your models, in vitro or in vivo, and whether the inhibition of ALK3 could have an effect on CYP2E1 activity.
Oh, thank you for the question. No, we couldn't measure CYP activities, and I think that it's really interesting and will give us a lot of information about how targeting BMP signaling is triggering the apoptotic and the antioxidant stress effects. But
Okay.
we don't know the technology, and
I mean, at least you can assess by Western blot. There is a very good antibody which is working on CYP2E1. If you want, I can give you the reference. And my other question is, did you compare the effect of DMH2 with N-acetylcysteine, in particular in vivo? Because there are numerous articles showing that different molecules have a wonderful effect on acetaminophen toxicity, but very rarely they compare between, their molecules with the N-acetylcysteine, which could be relevant because, it is the reference drug to treat acetaminophen-induced liver injury.
Okay, totally agree. We perform the in vivo model as a collaboration with Dr. Cubero, and just to check if this inhibitor worked. Being realistic, I didn't expect the effects that we did. I didn't expect that it works as well as it worked. I don't know if I mean. So, and we have a very little group of mice only, and that's why this is a proof of concept. And I think that it will be really interesting to do a study with different doses and perhaps with not N-acetylcysteine as coadjuvant, or well,
to compare.
really DMH2 as coadjuvant of N-acetylcysteine.
Okay. It's okay for the moment. I may have a comment after.
Okay, thank you, Bernard. Michelle, you also wanted to add something?
Yes, can you hear me?
Yes.
Okay, thank you very much, Águeda, for this very nice presentation. My question really concerns about, and maybe I missed this in your lecture or your presentation, but what is the dynamics of the ALK3 in time? So after an overdose of APAP, is there a profile? Is there an expression profile? Is it coming up and stays on long, or it's very short?
No, okay.
This is just to question you about the possibility using this DMH2 inhibitor for the ALK3, and in terms of practicality, if somebody has an overdose, and how long or in what time frame you should use this kind of inhibitors to ALK3 to prevent liver damage.
Oh, thank you for your question. The increased expression of ALK2 using DMH2 inhibitor, we observed it at the end of the experiment, and a longer time in vitro. But in vivo, we did not check the expression of the receptors. I know that it's something necessary, but the antibodies do not work in liver tissue. I don't know why. They work well. They don't work really good, but in cells we got a clear band, but in livers we got multi-band profile, so we could not check the expression at protein level.
Okay. Thank you.
Bernard, you said you wanted to make a comment afterwards. I don't know if you want to make it.
Yes, if nobody else is asking question. I mean, just I'm come back on your remark about apoptosis with acetaminophen. Actually, the acetaminophen does not induce apoptosis. It's only necrosis. There are very nice reviews made by Armut Jaeschke in the US. I mean, who is the world expert on acetaminophen, and it's even tunnel positive cells does not mean that it is apoptosis. It could be also necrosis. So I mean, you can read a very nice paper from Armut, who is giving all the arguments for to demonstrate that there is no apoptosis, but only necrosis.
Oh, okay, thanks. I will appreciate it, because I always wonder that if we have fragmentation of caspase-3 or tunnel positive cells, there is apoptosis process.
Yeah, but in this review you will see that different markers are necessary to tell that there is apoptosis. Just one or two is not enough.
Okay.
I mean, for the expert, I mean.
Okay, thank you.
Okay,
I'd say so,
I also, I'm sorry. I wanted to just to make a short,
well,
I'm not sure if it's even a question, but you wanted to ask something, Javier?
can you hear me good? Can you hear me?
We
Yes.
can, yes.
Okay, well, I was just going to comment on Bernard's
okay.
last comment on necrosis and apoptosis. I think, I mean, as a matter of fact, there are a few reviews, but especially papers, no, pointing that it's necroptosis, not only necrosis, because you have RIP kinase 3 knockout mice. They're protected against APAP, no, and this was published by the group of Kaplovich, no. And but at the same time, these in vivo experiments with the tools that Tom Lude has in Düsseldorf, they demonstrated last year in a paper in Immunity in cancer, no, not in APAP, that they have this RIP kinase 1, 3 phospho-MLKL and other markers. They demonstrated that with APAP, and I've seen some of the results, that you have activation of BCL-XL and the mitochondrial dysfunction together with the formation of the necroptosome. So it might both mechanisms be involved, not only one.
I think
Yes.
there are some different some different hypotheses and arguments for that. I think that's some people are fighting for that. I'm not an expert about cell death after acetaminophen, but I think there is some disagreement about that.
It's controversial because some of the inhibitors from necrosis, like necrostatin, only works at the beginning, but it's supposed apoptosis comes out up. So this might be, I mean, it might be worth studying at different time points after APAP and with different inhibitors. Yeah, I agree that it's very controversial still.
Yeah, and I agree with you that it depends mostly or probably on the model, because different type cell can be used. I mean, type cell, hepatocyte, but it could be HU, H7, it could be FG2, it could be HepIRG, etc., etc. And in vivo also, I agree with you that it could also depend on the timing on when you are looking for the injury.
I think this is very important as well in terms of what Águeda also mentioned, was this the role of ALK3 is. Águeda, just another question, quick question. Did you check the expression of the ALK3s on the ALK as well with other inductors of DILI instead of APAP?
No, we did not have the opportunity.
I mean, there are some other very nice model molecules that induce DILI, and that would be interesting to see what your pathways were doing there as well. So
For sure.
yeah, okay.
Actually, following this question, that's what I wanted to ask, because we see a compensation in the mechanism. When you inhibit LK3, you have an induction in LK2 and vice versa, right? So
Yes.
I think I was wondering, is it an effect of ILK3 induction or inhibition, or an effect of ILK2 induction?
Okay, thanks. I love this question, okay, because in one moment, one historic moment, we almost give up with this study, okay, because the expression of the receptors were so tight, and it's true that we see an upregulation in ALK3 when I inhibit ALK2 and vice versa. But it's true that this induction or this increase is very low. Okay, is 1.5-fold only. And when we overexpress ALK2, we expected to find the same the same result as inhibiting ALK3. But we only found the anti-apoptotic effect, but not the antioxidant effect. And I think that the modulation of ALK3 and ALK2, this little modulation is necessary to have the whole effect. If you only modulate ALK3 or only modulate ALK2, you don't have the whole effect. So you need to inhibit it ALK3 and overexpress ALK2 to have the whole expression. Because with LDN, one of the inhibitors that inhibits ALK3 but also ALK2, we don't have the same effects.
So maybe you will have to check upstream what is happening afterwards to see exactly.
Yes, I think that this is the key of this modulation.
Bernard, you have another comment.
Yeah, and no, because this acetaminophen it's crazy because there are numerous questions and it's very important for health and for physicians that are just facing this toxicity. Yeah, besides the CYP2E1, I wonder whether you look at the expression of enzymes that are responsible for the synthesis of glutathione, for example, gamma GCS, because to know whether the ALK3 could also act on this pathway, on the glutathione synthesis pathway.
Yes, we want to follow this investigation, deepening the antioxidant role of ALK3 or in the antioxidant effect of the inhibition of ALK3 and the anti-apoptotic role of ALK2 overexpression in other models. Because I think that perhaps in other liver failure models, but acute and chronic, we will observe better the separate signaling of ALK2 or ALK3 by using perhaps the ligands, the specific ligands of one or other.
Okay. You have a lot to do in the next years. Nice, very nice.
Yes, thank you so much. I don't know if anyone else will have any other question. If not, I think we will finish. We will thank once again Águeda for contributing for those seminars that are always very high level, and you kept it. So thank you so much.
Thank you
also for the organization. Thank you.
Thank you.
Bye.
Thank you very much.
Bye bye.
Thank you. Bye-bye.
Gracias a vosotros.
De nada.